from Microbe TV, this is TWiEVO, This Week in Evolution, episode 25, recorded on November 20th, 2017. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hey, greetings, Vincent, from the snow-capped Elde Lab Studios.
You got snow out there? Or?
We do. So just in the last weekend, got the first real dump, at least in the mountains. So between, 1 to 2 feet. Dry in the city now, but you can see that snow starting to pile up at higher elevations. It's a really beautiful time of year.
Yeah, no, as the winter progresses, you're going to have snow there in the flats, right?
We do, yeah. It kind of comes and goes and can accumulate. And then the real game, though, is just a half hour away at any of the mountain resorts where you get, bases that can go above 6-8 feet, something like that. And so that's where the fun starts.
Cool.
Yeah.
And we're at 25 now. That's like just over 2 years worth of podcasts, right?
Quarter century of TWiEVO podcasts. I love it. I mean, yeah, it's so much fun.
Still having fun?
Definitely.
Good.
Yeah. How about you?
I love it. It's different. It's different topics and different co-hosts. I love it. I love talking about science. Even if we only do it once a month, it's okay.
I agree. There's so much interesting ground to cover.
I mean, you and I swapped like 6 different papers for this one, right?
That's right. All of them could be easy keepers.
Yeah.
And I bet some of them we'll get to at some point. We also have, to get guests now, it's becoming more and more clear to me that sometimes you have to work ahead a month or 2 or even 3 months, which sort of changes my usual organizational style. So we've got stay tuned. There's going to be some really cool episodes ahead with some really spicy guests as well.
Yeah, we do on TWiEVO. We have to get people lined up sometimes a long time in advance. Yeah, because people have other things to do. So
yeah,
and also we have to get them a headset, and that takes time. Although that doesn't take months. That just takes a few days.
Yep, that's right.
It's always nice to have a guest because you can hear their point of view, right?
Totally agree. Although I think the nice opportunity that we have today is that, it's been a while since we've kind of hit the mailbag. Well, first just sort of knocked a paper off, just you and me kind of back and forth. But then we also have, a terrific conversation going with many other people who aren't guests, but they're writing in. Some past guests, some potential future guests, but other listeners that are contributing in big ways. And I think we're a little behind the curve in getting to some of those stories. So I'm really excited to do that today.
Yeah, we have to give them their moments because they do write and have good points and they're part of the conversation, right?
Yeah, totally agree. And so we do have a main paper that we'll cover today. I think it's really interesting. We'll get at some issues in population genetics and quantitative evolutionary genetics that we've haven't discussed for a while. But then we'll move relatively quickly through that and then dive in to the mailbag and see what's lurking.
Sounds like a plan.
Cool.
Now, this paper, how did you, it happens to be our third pigeon paper, is that right?
Yeah, this has emerged as a TWiEVO sub-theme. Anything with wings. We also have a number of fly and moth and related species in our archives, and we'll probably tap back into that a little bit. I guess, maybe part of this, as it relates to evolution, is that a lot of winged critters, insects in particular, and as we'll see for this bird today, travel or accumulate in massive populations. And because of that, at high, especially effective population sizes, we'll get into that concept a little bit. This means that there's a lot of time for sort of evolution to unfold. And that has some important consequences for thinking about the evolutionary processes. And so I think perhaps it's just that, both in the sort of charismatic species, beautiful birds and butterflies and everything else, But also there's, these are the places, if you're interested in population genetics, where it makes sense to do a lot of the research.
Of course, that's why we love bacteria and viruses, right?
Yeah, there's another example of some big population sizes. Maybe not quite as charismatic, though, right? It's hard to get the cover of a journal if you've got some fancy bird in the mix.
I don't know. I think viruses are pretty beautiful, especially if you color them in, All right.
Well, yeah, that's right. Well, you and me both, and you're kind of preaching to the choir. I think we've,
of course.
broader public might not be quite as quick to jump on the bandwagon of equating beauty to microbiology, but I think it is certainly true. The deeper or the farther we explore, the more amazing complex forms that are emerging in the microbial world that rival in beauty some of the more macro fauna as well.
I mean, the thing is that we have been able to study large populations of viruses and bacteria for a long time, but only recently have we been able to do this with larger animals because of all the advances in molecular biology that we've talked about, right?
Yeah, exactly right. And especially with genomics, in order to sort of have that, advantage or the usefulness of a large population in a species that shares a last common ancestor much more recently, or, in the case of viruses, it's not clear, sort of in the primordial days of life, what the relationships are, this, sort of controversial idea of fourth domain of life for the virus that we've touched on and things like that. And so these winged critters, and a few others, give you that opportunity to sort of take advantage of the large population sizes in species more closely related to us.
Yeah. Now, is this correct that this paper was sent to you last night by Steve Goff?
That's right. This is fresh, hot off the presses, and maybe sort of introduction to this episode where we're trying to respond to our great listeners and guests. So Steve Goff wrote us a quick email and said, hey, what about this paper that just sort of, I think, made a splashy appearance last week? It's on passenger pigeons. We'll talk about this in a moment. But Steve pointed out this really cool quote. So the paper was covered, I think, in the New York Times, and there's this quote about the work that the results are totally the opposite of what you would expect. And I think maybe that caught Steve's attention. This important sort of concept for biology in general, which is, it's the things that you are most surprised by that sometimes give you the greatest insights moving forward, the stuff that you wouldn't expect.
Yeah, for sure. We make assumptions often and we're surprised. Maybe that's what caught his eye.
I think so. Yep. As well as some interesting biology here. So the paper was published, I think, in last week's Science magazine. The title of it is Natural Selection Shaped the Rise and Fall of Passenger Pigeon Genomic Diversity. And the paper is from Beth Shapiro's lab at UC Santa Cruz. They have branded the lab as paleogenomics. Really well known for some of these ideas of things like species de-extinction, the woolly mammoth. This sort of push now to think about taking advantage of the ability to do genomics in species that are extinct, but where specimens or samples are available. This could include, archaic Neanderthal bones or frozen permafrosted woolly mammoth, or in this case, the more recently extinct passenger pigeon that we'll focus on in the next few minutes, to both do genomics and then think about the population genetics from that perspective of actually having kind of a footprint of what a snapshot of what the genome looked like hundreds or even thousands of years ago.
I'm just stunned that the passenger pigeon was once the most abundant bird in North America. Did you know that?
I think I'd heard something about that. I mean, there's that, there's the famous, this famous case of extinction where there were, this bird was really prevalent across all of North America. And then I think it was 100 years ago, that the last passenger pigeon at the Cincinnati Zoo, and I think her name was Martha, with literally no way forward to no male passenger pigeon, and she died. I'm reading here at the age of 29, and so
Martha, my dear.
That's right. And so Yeah, I guess what's really striking in this case is that compared even to other pigeons, as we'll see in a moment, that the passenger pigeon was especially prolific, and then went all the way to extinction, mostly at the hands of hunters and other, as our species started to expand and settle in North America in pretty impactful ways that ended up putting the passenger pigeon on a road to extinction.
So hunting was part of it? Is that right?
I think that was a big part of it. Yeah. I mean, you could imagine, of course, things like loss of habitat as forests or other areas were cleared. But I think really in this case, the biggest pressure on the passenger pigeons, I mean, it was if you look at some of the photos or images, it was a pretty charismatic and not a very well hidden or camouflaged bird. And so it became sort of a trophy or a staple to hunters among our ancestors who really went after it in a big way.
I suppose similar fate happened to, many species, but maybe one of the more prominent ones, the buffalo, right, of the American West.
Yeah, absolutely. And so the bison is starting to make a comeback. Yep. Fun for me, at least fun local story. We actually had I attended a bison auction about 2 weeks ago near Salt Lake. There's a state park that I might have brought this up on a very early episode of TWiEVO. There's a state park, Antelope Island, that manages a herd of bison.
Yeah, I remember. Yep.
Yeah. And so the cool thing there is, there's just enough diversity left and enough sort of ecological stewardship that they have these auctions between parks and ranchers and so forth. And so along with the genetics and the genomics to determine or measure whether these are sort of true breeding bison lines, that they're looking out for is that they're not introgressed with cattle genes. And then setting up, sort of a balanced strategy for mating and then bringing in genetic diversity, but keeping sort of the American bison going. So that's one that got right on the edge and at least in some circles, I think, has recovered or stabilized to some degree, whereas the passenger pigeon wasn't quite so lucky.
So the passenger pigeon's gone, is that right?
Yeah, correct. So I think Martha, that one,
that was the last one. Huh?
Bird in the Cincinnati Zoo, I think is sort of recognized as the last, if not the very last one, sort of symbolizes how back in the early 1900s, these birds were wiped out completely.
Well, now, since we have the genome sequences, maybe we could reconstitute one.
Yeah, so those kind of ideas. So this is hints at this idea of de-extinction, which at first sounds like totally crazy and wild. And I think that's certainly the case if you're talking about, kind of starting from scratch and doing a synthetic genome. But in the case of, for example, the woolly mammoth, and I haven't followed this field too closely, but one of the ideas is to actually take, existing species that maybe the closest, had the last common ancestor sort of the closest to any other species to the woolly mammoth, and then start to, raise them and breed them the same way we sort of do with bison today, but in a habitat that might more closely match the what the woolly mammoths were doing. So, obviously this would be farther north given climate change. And then I think, between sort of selective breeding, so the process, it's not going to happen overnight, but the idea would be through selective breeding, knowing what the ancient genomes look like, to try to sort of select sort of a hybrid approach, right, of using new technology, the genomics, as well as time-tested animal husbandry and domestication and breeding and so forth, to try to at least approximate in genetics the woolly mammoth, and then perhaps even as genetic engineering approaches get more powerful, maybe even making some key tweaks. So not doing the whole genome from scratch like that, but maybe adding in a key feature if the biology is well enough known and the engineering tools far enough along.
Well,
you never know.
I don't know if we need woolly mammoths walking around, though.
Yeah, I think it's interesting how that one sort of has come up in prominence. And in some ways, I think it's a combination of the timing of extinction. So it's a pretty massive challenge. It's obviously a very large species, but with some extant species that sort of maybe allow for a pathway forward. But I agree. I mean, I think you have to kind of consider the whole picture here. Like, what are we really? What's the bigger point, or what are we going after here in terms of?
Sure. Yep. All right, passenger pigeon is Ectopiste migratorius. 3 5 billion at their peak numbers.
Yeah,
highly mobile, bred in large social colonies, and lacking clear geographic structure. And they say few vertebrates have populations this large and cohesive. And that's the starting point for this study. Nels, they have
a lot of pigeons.
They have it's a lot of pigeons. They do a lot of sequencing of genomes of these passenger pigeons in this paper. Where do they get them from if the pigeons are gone?
Yeah, good question. So they do. I did a little bit of digging in the supplemental. So like a lot of papers in science, right? So you sometimes get a kind of almost an executive summary because the papers are so short. And then you end up, if you're interested in more of the details like this, you kind of have to go into the supplemental files. And indeed, so doing that and actually even reading the acknowledgments, my impression is that a lot of these passenger pigeons samples were from museums. And I think in particular, the University of Rochester Museum or the Rochester Museum, there's an acknowledgement to that group. And so we've seen,
another example of the power of museums, right?
Yeah, right. So we've kind of talked about this a little bit on TWiEVO before. We had Josh Drew on TWiEVO 12, Take a Left at the Goats. He was talking not about necessarily genomics, but even sort of using the museum specimens and where they were collected and when they were collected to make inferences about the ecology in places. Here, the authors and others are taking advantage of the preservation of some of these species in order. In this case, I think they took some bone samples to extract DNA from these museum pieces and then get a new sort of deeper look at the genomics of the passenger pigeon. And so this wasn't the first instance of doing this, but in fact, in the methods, the sort of complete methods, they describe, I think, sampling something like 80 birds in order to find the ones that were best preserved and then move forward with a smaller number. 41, they say that they actually did complete mitochondrial genomes, so not the full genome. And then just a handful of maybe a couple birds where they actually did go after the entire genome.
Now, they initially, in the very beginning, they initially reference a previous study of an analysis, a preliminary analysis of passenger pigeon genomes, and they make the statement, which this paper is based on, that they had surprisingly low genetic diversity for such a big population. Now, if you didn't know that was going to be the case, why would you sequence these anyway?
Yeah, great question. So the main reason so you're right. So there's sort of this paradox between these large population sizes, that 3 to 5 billion that people estimate in sort of census population, but low genetic diversity. And so by getting actually, more samples to have some comparison points, what you can do is start to apply various population genetic approaches or metrics or tests to start to try to get some insights into sort of what's going on here. Are the populations really that big? Is there more genetic diversity than we are currently estimating? And so by adding or filling out the data set, they're able to actually, as they're estimating some of these kind of key population genetic parameters, they're able to make a better case one way or another for what went on in the past of these birds.
Now, how would you quantify genetic diversity in a population?
Yeah, so there's several ways to do this. So, and actually first, since we've already begun talking about just population size, this actually ends up being a really key parameter here for the population genetics. And so, mostly we think about population sizes the way we've already been talking about it, how many total individuals are there in your population, the so-called census population size. And this is in the billions for this species, or was until they went extinct. But there's an important sort of second concept on population size, the effective population size, that population geneticists sort of pull their hair out about or really get excited about. And so this is an idea that actually I think was first proposed in the 1930s by a famous population geneticist from University of Chicago, a guy called Sewell Wright. And so the effective population size actually doesn't kind of count how many individuals. It instead thinks about so-called like idealized population based on things like the mutation rate, the amount of genetic diversity, And then these sort of concepts, and we won't go too deep into it, into all of the nitty gritty. It would take, a whole semester course or something like that. But this idea of identity by descent or coalescent theory, where you start thinking about when you think of any genetic variant in the genome of a individual in that population, how far back does it go? And actually, by thinking about the genetics this way, it's been a really powerful and insightful way to kind of advance ideas about how populations evolve. And so it turns out this effective population size, and it's calculated by different parameters, not by counting, that usually results in a much smaller number. And so that in the authors of this, using their new expanded data set, and in particular for mitochondrial genomes, do some calculations to suggest that the effective population size was in the millions. And that sounds small, but it turns out that's pretty massive. So I think, I don't have it, I meant to look this up, but I think the current estimate of our own population size, the human population size, might be something or in the neighborhood of like 50,000, 30 or 50,000.
Wow. Like I was going to ask you that. Yeah.
Yeah.
We have 7 billion humans on Earth, right?
Correct. Yeah. And so, this is where things like generation time, mutation rates, sort of contributions of individuals to overall population can really be a different parameter than the census population. And so that first sort of finding, and it's just, I have to say, it's not a completely new finding. So that estimate had been done for the passenger pigeon based on smaller data sets. And it doesn't change the view radically. But what it does do is it sort of shows you how this was, from a population genetic viewpoint, a very big population at some point in its past. And then this will become an important issue as we continue. Yeah.
Has this been done for other species? Do you know?
Yeah, it has for quite a few. So in fact, our winged friend, the Drosophila fruit fly, a lot of effort and energy has been put into the quantitative population genetics of those species. And there you have, kind of that further advantage of genetic manipulation and tools, obviously. So this sort of confluence around the 30s, actually, of population genetics with sort of classical genetics and then finally molecular genetics. The fruit fly is one of the major engines of this. And so, yeah, there's a lot known about that. And I think so the effective population size of, say, Drosophila melanogaster, I don't know if it's quite in the millions, but I think it is. It's quite high, especially compared to our own species. And so that again illustrates sort of how evolution might have sort of a very different sort of properties in species with super large population sizes compared to ones with small ones. So the way that natural selection is acting could be very different under these scenarios. And so that's why it's sort of worth it, I think, to consider these comparisons.
Nels, if I gave you a tube with 10 billion pox viruses in it, could you figure out the effective population size?
Yeah. So there, I think the main thing there is actually, the answer is no. I couldn't, because if the only information I had was that tube that contained 10 billion, and that's because I wouldn't know anything about the history of those viruses, right? Like how were they propagated? How did you amplify them? Was that all in one step, or was it sort of with bottlenecking over a period of time? And so that's a case where it could be very much true that census population size might be closer to effective population size, given what might happen in the next generation. How many of those viruses might contribute to replicating? Although you might see massive drops in effective population sizes with defective interfering particles or other things that can occur in virus populations. But this is a case where, I think the viruses do have some really important possible advantages because you can really control those populations almost completely. That sort of maybe balanced slightly by it's really hard to see kind of reproduction happening in real time because it's happening, maybe in an individual of another population that has sort of fluctuations in population sizes. So going back into deep time over hundreds of years or thousands of years for viruses is really challenging compared to these.
But you have this information about how you propagated them, what kinds of bottlenecks occurs. You would have that for passenger pigeons and humans to be able to
absolutely
calculate the effective size. Yeah,
that's right. And then you make estimates on things like mutation rates and several other parameters like this. And in some cases, these are estimates. And so I think you have to take any of these numbers with a pretty big grain of salt, because there could be new data that emerges that we are measuring the mutation rate in ways that weren't really accurate. And so this is it's not kind of you define this once and you're done. It's sort of an ongoing process where we try to get better and better in our estimates as we have more information to inform that biology underlying these parameters. Yeah, that's right.
Why did they initially just look at mitochondrial genomes of these birds?
Yeah, so remember, these birds aren't super old. They're probably collected over the last several hundred years, and probably many of them during that sort of boom in hunting and collecting. And so, but these still are hundreds of years old in many cases. And so they kind of qualify under this category of being ancient DNA. And so, so when you're collecting ancient DNA, and there's been just huge, really exciting advances for like Neanderthals, Denisovians, other archaic human or Homo species in our own genus. But one of the big things that has sort of led that forward is being able to deal with the errors or contamination that can arise if you're not careful with these samples, right? So the biggest issue for sequencing our own species is that we have our own DNA kind of that we're spreading all over the place. And so how do you ensure that you are tracking correctly not only the sample you think you are, but that as the DNA just starts to break down or even mutate over time, that sort of process of degradation, that you're not just sort of sequencing a bunch of errors, that you're sequencing. And so in fact, that in the main body of this paper, they don't devote any attention to that. But if you go into the supplementals again, there's a pretty large section on how they try to ensure that there's not a lot of error rates. So taking that into account, then you can imagine that it's a lot easier to get a handle on the much more modest mitochondrial genome relative to the entire genome. And so there you're kind of prioritizing, right? So they're already making decisions on what are the best samples in terms of integrity of the DNA.
Right.
And then from there, from the best ones, first of all, doing mitochondrial genomes, but only in a couple of cases do they go sort of all the way through with the ambitious goal of sequencing something like the whole genome. And so, that actually works out okay. You get the mitochondrial genomes give you some information. You have the advantage that the mitochondrial lineage is passed only through the mother. And so that actually for population genetics can be a useful sort of benchmark or comparison point to other studies. But if you're kind of thinking about the genetics as broadly as possible, having the entire genome has some advantages too, because you can really kind of get under the hood of the evolution that's happened over hundreds or thousands of years.
Okay. And they used as a comparator for the genomes a related pigeon, right?
Yeah, that's right. So this is the band-tailed pigeon. And so that's the sort of one of the existing species that didn't go extinct. That's probably the most closely or close to the most closely related non-extinct relative of the passenger pigeon. And so we did, if you remember, we did a TWiEVO with one of my colleagues, Mike Shapiro. This was back a while ago. He uses other breeds of extant pigeons. This was TWiEVO 7. But is using them not as much for a population genetics sort of comparison point, but the developmental evolution of all of these sort of derived traits, the feathery boots or the weird head crests. And so this is another case where the pigeons show up to be an interesting biological comparison point. So in this case, the bandtails were useful because of that somewhat close relationship to the passenger pigeons. But even more than that, what the authors point out is that the bandtails are believed to have had much smaller effective population sizes compared to the passenger pigeons. And so that gives them a comparison point to sort of think about what this means for genomic diversity and as a comparator.
Mm-hmm. All right, so they figure out a parameter called nucleotide diversity, which they give a symbol pi, right?
Yeah, right. So there's then a few, kind of metrics. So we already talked that population geneticists get kind of excited about and are into. So, one of them is effective population size. And then there's also this idea of genetic diversity. And so, and whether or not sort of the mutations that you're looking at are being acted on by natural selection, or this idea of sort of Darwinian survival of the fittest territory, so to speak. So, and this all builds out. I thought this would be a great chance to just touch up on maybe a couple of just the sort of core ideas in population genetics. And so, because this kind of builds for all of the metrics or the tests that they did with that comparison between the band tails and the passengers. And so, the sort of kind of one of the central pillars of population genetics for the last 40, 50 years has been the neutral theory of evolution. This was pioneered by Mutu Kimura. There's also sort of an offshoot of this, the near neutral theory that came some years later and after some debate by Elde. And so this is the idea that actually most variation that you will sample, the genetic diversity, the variation that you sample in populations, is not a result of natural selection. It's not this kind of survival of the fittest space, but instead that most mutations are either are neutral or slightly deleterious, and then can fix or can sort of rise in populations or accumulate by the process just of genetic drift. And so drift, of course, is this idea of sort of this randomness in as populations bottleneck, that there's going to be some variation, and whoever has kids, their variation will go forward. And that's not always because they outcompeted everyone. It's just they had kids, and this was the subset of variation. So the kind of grabbing a marble from a bag analogy is commonly used, that there's some chance in which marble will come out of there. And there's also some chance in who might make it to the next generation. And so the idea of neutral theory is that that's actually kind of on average, or in the whole picture, that's the main way that genetic diversity propagates. And this has been pretty contentious, actually. And part of it is, I think, if there's this so-called neutralist versus selectionist debate. Part of this kind of hinges on how a lot of us grew up as in our own training in biology. So a lot of, I would say, experimental genetics or molecular biology, molecular genetics involves, this idea of the genetic screen, or that you're measuring phenotypes, things that have had a consequence for fitness in whatever experiment or whatever observations you're making. And so that is sort of perhaps colored our view. I mean, I count myself in this category. I grew up as a cell biologist, that all of evolution, is sort of there almost for a reason, or it's been selected on, sort of again through that Darwinian viewpoint framework. Where in fact, the population geneticists for more than 100 years now have been saying, hold on, it's certainly true. They're not the most proponents of neutral theory are not at all saying that selection isn't there. It is there, but those are the rare cases, actually, if you look at the whole picture. So it's this really interesting, almost historical or certainly historical, almost cultural divide, I would say, between evolutionary and experimental geneticists. This idea of how is selection acting or not acting on populations.
It seems to me that this is like contrary to this whole idea of quasi-species and selection of RNA viruses, right? There's enormous diversity, and you have selection that pulls out certain sequences from that.
Yeah, absolutely. And so there, it's in that case, you can imagine that those viruses are under pretty strong selective pressure by, the immune system, right? The diversified immune system of the cell. And so because in these sort of genetic conflicts that kind of underlie the interactions between viruses and their hosts, that is probably a case where natural selection can, certainly for virus evolution, might dominate in those cases. Although there again, depending on population sizes, depending on how well adapted the virus is to the host, then and certainly depending on the dynamics of infection. So, let's think about, for some of these quasi-species, right, that go through two different hosts, the arboviruses that go between insects and, say, a mammalian host. If there's a huge bottleneck there where, you might have millions or even billions of particles that sort of get down to a few particles that make it, there's actually a lot of randomness in that bottlenecking. And so that will influence, And so so even in cases where there's clear sort of dominating natural selection, you can certainly imagine situations where drift will have a potentially, pivotal outcome on how genetic diversity propagates through those populations.
Yeah. I always tell people it's like, even if you have a mixed population of RNA viruses and you select on some gene sequence, you're going to have a lot of passengers going along with that. And that's pretty random, right? Right.
That's right. And so that's a related concept of genetic hitchhiking, which comes up as well. Yeah. And one point to make is actually, sometimes we think of drift, it's almost just kind of intuitively, or this idea of neutral theory or neutral evolution, that means that there's no selection at all. That's not quite right, actually. And there's when you think about it, like these mutations, like say it's to a gene that encodes like an important component of the ribosome. Mm-hmm. If that's like a mutation that's a null mutation or a knockout mutation, that's not going to make it, because in that case, if you have a debilitating mutation, you can't sustain that in a population. And so that's kind of an important, subtle, but I think important concept, that even when we're talking about neutral theory, the variation we're talking about, it still is, if it's in a gene, for example, it might be a variant in a ribosomal protein, but it's not one that kills the entire ribosome, because that just is sort of a non-starter. Yep.
Now, one last point here. The term drift, it's used differently depending on the situation. You've been using it in one way to denote non-selected or somewhat non-selected, or you just said it's not always not selected, changes. But it's used in influenza virus to describe the immune-mediated selection from year to year of a virus. Antigenic drift.
Antigenic drift.
That's different because it's totally selected, right?
Agree. Yeah, I agree. And we run into this, these terminology differences can trip up entire fields sometimes. The difference between epistasis actually has different definitions in some genetic circles than it does in some evolutionary circles. And so it can be a little bit tricky as you dig in. So, I agree. It's important to kind of take these things into account as you're thinking about this. So anyway, getting back to these passenger pigeons, then just as you're saying, they wanted to look, so they measure genetic diversity, that pi statistic, and in fact, they also measure a couple of other sort of important population genetic metrics, and looking specifically at, or trying to infer sort of the efficiency or the prevalence of adaptive evolution. And so they measure something called omega A. This is similar to a DN-DS ratio. So these are the rates of non-synonymous to synonymous or silent codon substitutions. And so this concept for population genetics derives from the degeneracy of the genetic code. And so, as I'm sure most of our listeners are familiar with, if you think about the DNA to RNA to protein, and the unit of the codon to code for an amino acid, that there's more than one codon per most amino acids, and 3, for example, stop codons in a lot of mammalian species that will all stop the extension of peptide. Mm-hmm. And so that allows you to actually, this has been a hugely sort of influential way of thinking about how evolution is acting on genes, is to compare the types of substitutions that are emerging for whether or not they're non-synonymous. That is, you change the DNA nucleotide and that also changes the amino acid, versus whether they're synonymous or silent, where you change the nucleotide, but the amino acid stays the same.
I was going to say, you and your trainer, Harmeet Malik and Mike Ammerman,
Sure.
This is the whole field of paleovirology is based on this and finding residues that are apparently positively selected, right?
Yeah, correct. And so there, the idea is that and it's a major assumption, it's an assumption that's violated from time to time, but is that a silent change is really silent, that you can change, if you are changing a nucleotide that doesn't change the amino acid, that won't have a massive functional consequence, because, at least from the protein level, the the amino acid within that peptide will be the same. And so it's silent, at least as defined that way.
And that would be drift. That would be drift, right?
Well, it would be, so actually another concept that comes out of this, and we'll talk about this, is purifying selection. So it might be that, so the idea here is that these mutations are tolerated. Yeah, you're right. So they're neutral in the sense that if you have if you make that change, it's not going to be selected on, it's not going to be purified out of the population because it's tolerated, basically.
Right.
And then it could be subject to drift, just as you're saying. Whereas for those non-synonymous changes, and this is where the ratio will become important, the sort of working hypothesis from the neutral theory of evolution is that if you make an amino acid substitution, on average, these are going to be slightly deleterious. And so what happens is they're purified or removed from the population. So this concept of purifying selection, what happens is your rates of non-synonymous substitutions, the so-called DN part of the equation, is really low, whereas the DS part of the equation is high. It's just basically a molecular clock. As mutation happens, and as these things sort of continue to happen over evolutionary time, you'll see more of them because they aren't negative. They're not purified out of the population the same way that a lot of the non-synonymous ones are. So then where the evolutionary virologists like Mike Emmerman and Harmeet Mullik get excited is when they actually see these elevated DNDS ratios, where now it's as if there's an overabundance of non-synonymous mutations. And this turns out to be diagnostic of so-called positive selection. There's many flavors of this. This is just one of them. But situations where in protein evolution, there's an advantage to that substitution. And so these cases of positive selection are really interesting, not only for sort of host virus or host microbial conflicts, but can even underlie some intragenomic conflicts involving things like centromeres competing against one another, mitochondrial genomes potentially competing with nuclear genomes, and all kinds of interesting biology, reproductive success, things like sperm competition. A lot of the genes encoding the proteins that dictate these functions are often evolving really rapidly. On the other hand, if we think about most of the genes, the vast majority of them have low DNDS ratios. And this is sort of echo or highlights gene conservation. The fact that you can pull the same actin gene out of, say, a budding yeast or our own cells and clearly see, even just by comparing the DNA sequences, that it's the same gene encoding for the same function. Those things have been conserved, in some cases, for hundreds of millions of years. And this is the basis of, why are model organisms so useful as ways of understanding our own biology, that we still share a lot of the same, not only biological functions with our common ancestors, but even that they're conducted by the exact same proteins that have been kept around under purifying selection for a really long time. So here the authors wanted to measure and compare, actually, whether they saw any differences between the passenger pigeons, given that they had these huge effective population sizes relative to the bandtails, and so they did a couple of flavors of looking at DNDS. So one is this omega A value, which actually considers sort of neutral, adaptive versus neutral substitutions. And then another related ratio, the PN to PS. And this is very similar to DNDS, but instead of fixed changes between species, this is polymorphic changes within species. And since they now had, 40 mitochondrial genomes from the passengers, they were able to make these calculations where you're asking basically among a single species in that population, is there polymorphism or genetic variation that is different within that same species? And so that gives you sort of yet another look at some of these population genetic sort of processes. So, and what they found, and this is shown in figure 3, is that the passenger pigeons had a higher so-called omega value that they infer to mean there's more efficient positive selection. They're correlating this to the large population size, whereas they also had a lower PN to PS, the polymorphism or the polymorphic measure of polymorphic changes, which implies stronger constraint in the passengers relative to the bandtails. And they did this by actually, most of these calculations were done for the full genomes that they had, where they calculated in 5 megabase windows these values across as much of the sort of contigs that they could assemble from them. And then if you really squint at these at the values in Figure 3, they look pretty similar to me, but they say that there's a subtle statistically significant difference between them.
Are any of these results surprising? Is it surprising that PNPS was lower in the passengers?
Yeah, I don't think it was super surprising. So the sort of population parameters here are very much consistent, I would say, with the population sizes. So then, actually, I think the bigger point of the paper comes from this last sort of point, which gets back to that first concept we were talking about. And that is this idea of the effective population size. And so what they, and they calculated this earlier to that, I forget the exact number, somewhere in the millions. And the difference here, or the thing that they point out after thinking about, they also actually spent some time of the paper accounting for GC bias. So this is during gene conversion, as the genomes are evolving, the base pair composition, the ATs, Cs, and Gs, that can change. And so given the large population size, I think probably in review, the issue was raised as to whether some of the metrics they're calculating were just sort of a side effect of a greater GC bias. And so they try to account for that a couple ways, and then kind of get back to the idea of this effective population size. So in previous studies, or sort of the kind of state of the art in the field, the idea has been that we know the passenger pigeons are extinct today. We also know that they have a large effective population size. And so what had been previously proposed by other population geneticists was that population size had wild fluctuations in the recent past, and that this might account for the vulnerability for the passenger pigeons to become extinct. And here, what the Shapiro group proposes is, and what they document from their comparison, is that in fact, and this is in Figure 1, is that in fact, going back thousands of years, they're inferring a relatively stable and consistent large effective population size. And so that's really, I would say those other parameters are certainly consistent with large population sizes, but this idea that it might be stable, such that, you are kind of assumptions. And this is that part, that great line that Steve pointed out from this article about the paper, is that sometimes you get a result that's totally the opposite of what you would expect. And so the fact that if that inference is correct, these things really did have large effect, if the passenger pigeon had large effective population sizes, we might expect it not to be as vulnerable to extinction, that you have natural selection acting efficiently in the population. So in which case that you would imagine that there's all kinds of diversity that could buffer sort of environmental changes. Even if part of the population is getting wiped out, there'll be other variants in that population. Maybe their behavior is different. Maybe their camouflage is different, such that they'll still somehow survive this massive pressure by hunters. And then those will be sort of the passenger pigeons of tomorrow. And so that ends up being, I think, kind of the final conclusion of the paper, is that in fact, well, and it could be sort of a depressing conclusion, is that our own species can alter the environment so drastically, in this case with a very acute pressure of hunting, so that even if you have what looks like a genetically healthy, or like a grade A report card as a species, that you're still vulnerable. And in fact, they even go one step farther to sort of speculate that because the population was so large, that efficiency of selection could actually become, turn into a disadvantage, where the population is sort of so well tuned to its stable environment that when you put in something that perturbs it, the whole thing is vulnerable and it all crashes. And that, I think, is sort of the kind of new big idea from this work that they infer from the comparisons they make.
So basically, they say, the diminution of their population by commercial harvests, these would be selection pressures that the pigeons never experienced in their evolution, and therefore their population size couldn't deal with it.
Yeah, or the implication of this would be actually the sort of paradoxical idea that if the population was sort of medium-sized, that it would still be at a state of sort of both genetic diversity and sort of related measures where they might have had a better chance of surviving, actually. And so I was trying to think of an analogy for this in, something that might be microbial, actually, given our interests.
Yeah.
So think about, there's this interesting history of attenuated virus strains that make up vaccines. And so what virologists have done, like my favorite example is in the vaccinia, the smallpox vaccine, where in the 70s, lots of virologists would passage hundreds of times virus populations in, say, chicken cells. And what you end up with are viruses that are acutely adapted not only to like chicken cells, but they have to even have the exact right media for the chicken cells for that virus to propagate because it's adapted so well to that environment. And so then you take that virus that you almost have to coddle because it's adapted in such specific ways to the host, which is the chicken cell with this exact concentration of serum even. And then if you take it out of its environment and use it as a vaccine, our full diversified immune system just crushes this thing. And there might be a case where even if those viruses had been brought through in these massive populations, that they might be more vulnerable to extinction when you do something like expose it to our immune system, or in the case of the passenger pigeons, expose it to our proclivity to hunt these things in big numbers.
Yeah, I like that analogy. So, Nels, if you took that virus, which now is over-adapted, and you treated it with a mutagen, you think that would improve its chances of moving to a different system?
Yeah, interesting. So I love this. And this is where, so the passenger pigeons are fascinating because this is, kind of our ecology. It's part of our natural history. But the magic of, thinking about this maybe in a viral system like you're talking about, Vincent, is that we can manipulate it and adjust parameters. And I would love to do those exact experiments, right? So you could hit it with a mutagen. You could, pay close attention to the population sizes, do the bottlenecking in different ways, start to control for some of these population genetic parameters, and then see whether you end up with populations that indeed are more vulnerable as you change environments. So I think that could be kind of a cool, hybrid approach of thinking about our natural history, but then doing experimental approaches as well.
Could you look at this issue, both your vaccinia example and the passenger pigeon, as one of these viruses and animals being in a sequence neighborhood that they can't get out of?
Yeah, so kind of thinking back to our last episode, right? TWiEVO 24, Good Viruses in Bad Neighborhoods with Marco. Right. Yeah, that's right. So I think this is sort of what's really fun about this, kind of our motivation for doing TWiEVO at all is colliding some of these classic ideas in population genetics with some tractable experimental systems. And so start thinking about not only these exact parameters we've been talking about, but also how that relates to the topic we talked about last time, which is the idea of these adaptive landscapes in a sense where the codons that are in that quasi-species or in that viral swarm, how those positions might also influence robustness or the fragility of an entire population. So you can imagine even taking it into sort of other directions along those lines.
Yeah. So in the end, this paper, it basically tells us that what we thought was the reason for the extinction of these pigeons isn't right, and now it could be something else, which is that it was hyper adapted to its particular environment, but we're not really sure. And it doesn't prove it, right?
I agree. I agree with you. And so, I wouldn't go too far with a definitive conclusion, but I do think, that it did gain some attention because it really, I think when we think about sort of what are the species today that are the most vulnerable, and in fact, this is certainly sort of disturbing trend line that as our environment is changing rapidly now and possibly hitting some turning points. And there are now it's already happening where lots of species are going extinct kind of all around us. I think some of the assumptions that are common, the intuitive assumptions, are that when you're at really small population sizes, when inbreeding is kind of necessarily high, and where we're depending on zoos and other sort of last-ditch conservationist efforts to try to rescue or save species from complete extinction, that in fact, we might be ignoring sort of an important trend on the whole other end of the spectrum, where a population that we would look at, like let's say an insect population that we might think of as non-threatened or really abundant, could they be vulnerable to something like this? And I don't remember the exact reference, but I think there was a study that just made a splash maybe about a month ago, suggesting that in some wilderness areas, I think in Germany, that insect species diversity is down something like 70%. And so are there cases like this all around us, or, right under our nose, but things we aren't realizing, things like microbial populations? Are they sort of rising and falling from these massive populations and then getting wiped out in ways that can support entire ecosystems? I think we have to be really open-minded to that there could be some real issues sort of on both sides of the ledger here.
Yeah, I think that's a good point. Even with great numbers, you're not necessarily going to be safe.
Yeah, and so certainly I think an opportunity for more research in that area. And I think the fact that it was so almost counterintuitive or totally the opposite of what you would expect is what kind of makes this important point to get out there.
Yeah. All right, Pigeons show the way.
There we go.
It's a nice paper. I liked it.
Yeah,
we had the opportunity to explore some interesting genetic ideas, right?
Agree. Yep.
How about some email?
Let's do it. So again, that was that Steve Goff just wrote us last night and suggested that paper. And maybe before we get into the mailbag here, just to say we are getting more and more other suggestions for work. And I just wanted to mention that I really appreciate it. And we're constantly getting great ideas. Some cases we've actually used those ideas. Other cases we hope to still get to it. It's obviously a fast moving target, but please, don't hesitate to make suggestions to us. We see them and we definitely consider them. And so, all ideas are really welcome, and we really appreciate it. So we also looks like we have some suggestions here that we should get into. Yeah. So maybe I'll do the first one here. So this is Kareem. He's referring to an episode we did actually on the pox viruses a while back when Rich Condit sat in with us. That was really fun. I don't remember offhand the exact episode. But so Kareem writes, "Hi, Rich, Nels and Vincent. First of all, I love your show. I'm a very faithful follower of TWiEVO and TWiEVO. Please keep up the great work. Just a little note that comes out of my slight disappointment. You give all the credit for the IRES RAC1 connection to a Sarah Cherry genome-wide screen paper in which RAC1 is not even mentioned in the main text. I love this. This paper identifies 66 ribosomal proteins, which is composed of roughly 18 proteins, as important for IRES translation. Please check my paper that formally proves that RAC1 is important for IRES translation. The reason of my, in quotes, childish disappointment is that I am the first author on this paper and spent 5 years of my life working on it. Happy face emoticon. No big deal or hard feelings, though. I know that all of us can miss stuff in the literature and are more than likely to remember faces and talks we see at conferences. Again, I love your show and will always be an ardent follower. Thanks for all your efforts and work making my commute from San Francisco to Stanford every day more pleasant." Well, listen, thanks, Kareem. And I would say, Vincent, I'm sure you would agree that we are always making sort of oversights, mistakes, slight blips. And so sort of in the heat of the podcasting battle, I would say sometimes we fall a little short. And so, Kareem, no, disappointment necessary here. You're 100% right. And I was just looking at Kareem's paper, I think published in Cell from 2014. And it really does look like a great study, kind of definitively bringing that connection between RAC1 and the IRES. So thank you for filling in some of those important details along that way.
And part of the problem was we just looked at the paper itself that we were doing, that POX paper, and they referenced the cherry screen, and we didn't go beyond that, So
correct. Yeah, so we can pass this one on to the author.
It's funny because the paper said that this paper identified, RAC1 is important for iris, and they referenced the cherry paper, but that wasn't quite correct, right?
Yeah, exactly. Well, and this happens all the time, right?
Oh, yeah.
There's, as authors ourselves, where cases where there's both a vast and deep literature, and you sometimes get it right, you sometimes miss a few things, and in fact, it's almost impossible to get everything in the picture since there's so much great and overlapping work. But I agree, Kareem, in this case, your paper deserves some credit
for sure
along this line.
No, it's a very nice paper.
Yeah,
and its title is RAC1 Controls Iris-Mediated Translation of Viruses.
There you go.
It's Cell 2014. And it looks like all the work was done in France.
Yeah.
I guess, So Kareem was at Strasbourg at the time. And now he's in Stanford, California.
Yeah, sounds like it. Thrilled to hear that you're a faithful listener. I really appreciate it, and hope that we're brightening up those commutes, Kareem.
Next one's from Jeff. Dear Nels and Vincent, I greatly enjoyed the recent podcast on Cnidarian dinoflagellate symbiosis. I was struck by your speculation on possible evidence for cycles of cooperation and conflict between symbiotes based on diversification of proteins associated with mediation of the relationship. Do you know of any specific examples of such cycles of cooperation and conflict, or references for investigations along these lines? I'm just getting into working on modeling of evolutionary dynamics, far from my background in experimental physics, and TWiEVO is proving an enjoyable and accessible introduction to the fields. And Jeffrey is at the Simon Center for the Study of Living Machines in Bangalore, India. You know any examples of this, Nels?
Yeah, good. Thanks for the letter. Really glad to hear that was stimulating. So, I would say a couple things, I guess. So one is on the Cnidarian dinoflagellate front. My sense is that is kind of on the cutting edge in terms of thinking about it, sort of these cycles of cooperation and conflict this way. I would say there are a couple of places I would look, though, or think about. So one is, so there are a few meetings that are specifically looking at this idea of cooperation in a few systems. So I'm thinking about actually bobtail squid. This has come up on TWiM, hasn't it, Vincent, a few times? The symbiotic, yep.
For sure.
Yep. So Margaret McFall-Nye's work, and a lot of labs that have emerged from her really great system over the decades. I'm also thinking about the symbionts of really recent symbionts, actually, of insects. And so this is sort of broadly speaking in the wheelhouse of Nancy Moran, but also labs that have emerged from her group as well. So there's actually a local connection here in Utah, a guy named Colin Dale, who looks at sort of, so a lot of folks are studying now in many different systems symbionts that have been around for a long time. So obviously, the most famous or celebrated of these would be our mitochondrion, I would say. But there are ones that are actually very recently acquired, even just sort of in the last blink of an eye, evolutionarily speaking. And so Colin is actually onto a few of these where they have the free living bacterial forms that are really closely related to very recently acquired symbionts of some insects. And it's a really funny story where Colin got a phone call from a pathologist here at Utah who said, we got this sample from someone who impaled themselves on an apple tree branch, and we cultured out a bacteria that looks like one that you just published on last year. Are you interested in looking at this? And and he's like, yeah. And so it turns out this actually allowed him to sort of pioneer the system, which is sort of a case of symbiosis just right on the edge, where you have a very recent transition. And so I think that's where you can imagine just sort of these cycles of kind of going between the two. But a good question. I'll keep my eyes open and maybe update you directly, Jeff, if we come up with something more direct. But good luck. I think it sounds like a fascinating thing to model and think about. There's some incredibly interesting biology here in these interactions.
More than we know. I mean, there are just a relative handful of known symbiotic relationships, but there's just tons of them out there.
Totally agree.
And I would argue that many viral infections are essentially symbioses, right?
Yeah, absolutely. There's actually there's a great viral system, so poxvirus system even, and I'm blanking on her name right now. She's down at Georgia. Also came out of Nancy Moran's lab, and she has a system where it looks like a poxvirus was recently domesticated in a sense by an insect that then uses the viral, proteins in some way to turn down the immune system to allow the larva that the insect injects into the host to develop. That's another really cool system where it's a virus case and where it's very obviously advantageous for the virus to be deployed this way. And there's many cases of that have been more long established. What's interesting about this one is that it's very recent.
All right, you're up next.
Okay, so Daniel writes, "Hi, Vincent and Nels. I have a cool suggestion for a future TWiEVO episode. This is a paper published in August in Science called Structure of Histone-Based Chromatin in Archaea. Work done by an amazing postdoc in a lab in my building, Francesca Mataroli, working in Carolyn Luger's lab at the University of Colorado Boulder. Cheers, Daniel." Very cool, Daniel. I think this one just briefly came across my radar, probably back in August. And I agree. I mean, this idea that, so first of all, I think just when we think about bacteria and archaea, that we sometimes almost intuitively underestimate how complicated and how sophisticated the biology is in these microbes. And as people are sort of peeling back the layers of the complicated and in some cases convergent, in some cases divergent core functions that are often only associated with eukaryotes, this is right in the wheelhouse, so to speak, of TWiEVO. So thanks, Daniel. We'll definitely take a look.
This is very cool. Basically, they figured out the crystal structure of an archaeal histone DNA complex, and they show that the geometry is exactly the same as in the nucleosome, which is, of course, the eukaryotic DNA histone complex.
Yeah, isn't that wild? Some of those deep threads.
Amazing. Of course, archaea predated eukaryotes, and so that means they'd already learned how to wrap up their DNA. It's just great stuff, isn't it?
Yeah, totally good. Yep. Or there are also many cases actually where surprisingly, over these long evolutionary time frames, that different systems converge independently, but on the same functions. And so you also see cases like that as well, where given enough time and sort of just enough of the same kind of biological constraints in a sense, or even, enough similarities in the environments, that evolution, in a sense, figures out an independent path using, in some cases, different tools or the same tools or hybrid combinations.
Yeah.
And so, I love these deep comparisons.
The next two were picks, so I'm going to move those down to our picks section.
Oh, sounds good.
And that brings us to Adam. Dear Nels and Vincent, as usual, great discussion and conversation during the most recent episode, which is no longer the most recent, of course.
That's right. So this is the DHX 9 story, just a couple episodes ago now.
Immediately after hearing about the RNA helicase DHX9, I thought of the 2013 PLOS Pathogens article from Grant McFadden's lab entitled Myxomavirus Protein M029 is a dual-function immunomodulator that inhibits PKR and also conscripts DHX9 to promote expanded host tropism and viral replication. In this work, the authors found that DHX9 interacts with the E3 ortholog of myxomavirus to promote virus replication under certain conditions. As Nels knows quite well, E3, encoded by the E3L gene named from vaccinia virus, is a double-stranded RNA binding protein. It is enticing to hypothesize that E3 recruits DHX9 to viral double-stranded RNA to help separate the strands and hence reduce immune stimulation. The authors did not directly study this in their work, so I'm only speculating. I very much enjoyed hearing about another interesting role for DHX9. Thanks for your wonderful podcast. Wow. That's an interesting idea, right, Nels?
It's really interesting, yeah. And thank you, Adam, for your wonderful letter. I love this. So, so this was episode 23. The ALUs are going to be all right. This idea that DHX9 is sort of the authors propose that one of the things that's pivotal things it might be doing is managing double-strand RNA that emerges from the transcription of ALU elements that are kind of all over the place, interacting potentially with circular RNA, other sort of moieties that you would want to keep managed in order to tolerate the presence of ALUs at all. We were kind of speculating at the end of that episode that this could also play perhaps a role in viral surveillance, right? So that you need to kind of keep the cytoplasm quiet in a sense so that you could then recognize as viruses come in and start replicating and making double-strand RNA. And so, of course, then you might imagine, and this is great, Adam, to make this connection, that the viruses themselves would have a way of trying to counteract and sort of fool the cell into thinking that it was not infected. And so, or to mask or hide the double-strand RNA or related molecules that could otherwise sort of sound the alarm for intrinsic immune response. So I really appreciate it. That got me thinking about some ideas that we've been thinking about along these lines. And so this is really helping to stimulate some conversations. And in fact, Grant McFadden, maybe not surprisingly at all to you, Vincent, is back at it again. He just published another paper in, I think, Scientific Reports. The mid-November issue just came out, and this came across my radar the last day or two. Identification of host deadbox RNA helicases that regulate cellular tropism of oncolytic myxomavirus in human cancer cells. And so,
is DHX9 in there?
DHX9 is in there. It is. So it's interesting, right? So they, again, using myxomavirus, and in this case, Grant has really been involved in advancing it technologically and medically as a oncolytic therapy, developing that. And so there's this idea that if you just look at how myxoma, which usually infects rabbits, might act in cancer cells, it's usually quite permissive, but not in every case. And so they did some library screens, knockdown RNA library screens to see what are the proteins. And specifically, they focused on the helicases, I think, given this earlier work that Adam is pointing out. And then asked, when they knocked them down, kind of one by one, all of the helicases, which were the ones that then, when they were gone, allowed the virus either to do better or, on the other hand, made the virus worse. And so they identified some in each category. And so kind of an interesting further connection about the interplay between viral proteins, these dead box helicases, and the sensing of double-strand RNA.
I don't know, Nels, maybe you should see if there's some virus host conflict between each E3 and DHX9, right?
Absolutely. It's right. It's on our radar. I think that could be a really cool thing. And it's part of my sort of evil plan that TWiEVO is going to drive forward how I do biology. I'll just wait for people to write and point me in the direction of cool stuff, and then that will sort of drive our own research program.
It's not not a bad idea.
Not a bad idea.
We've got some very good suggestions here.
Totally agree. And making those, this is the magic, I think, of having these conversations, is to draw these connections in new and different ways where we're all kind of spotting things, thinking about it differently with our own different experiences. I mean, this is a great way to drive science forward. And so this is why these conversations, I think, are so important.
No, I totally agree. And that's why publishing them is key, right? You and I have a chat.
Yeah.
And if it just happened in your place, it would be fine. But when we involve as many people as listen, then it can get incredibly powerful.
Yeah, great point, Vincent. And so the more ways that, I think we sometimes think about science outreach to the public, but actually science outreach to other scientists, whether that's through kind of traditional means of publishing in peer-reviewed journals, if that's pre-printing on things like bioRxiv, or even if it's doing these sort of, kind of offbeat things like podcasting, or even just communicating our science to the public, but with scientists who do super different things. It's a new way of kind of causing these collisions of ideas that could really lead to cool stuff.
Yeah, I agree. I think it's very powerful, and I think it's underappreciated by most scientists. I think the public loves it, but I think scientists are still, in their usual ways of communicating. They mostly read and give talks, right? But this concept of listening to other conversations, they just don't, I don't think they get it, but it's what they do every day,
Yeah, and to share that even more broadly. That's right. Yep. Bring a new level of inclusiveness into our communities that way. I think it can be really important. I also, on the other hand, as sort of a, shy scientist from Lake Wobegon originally, it can take a little bit of courage or a little energy to get up the courage to talk about stuff that you might not be super familiar with, or to try to think about communicating it to a group that you might not, where it's difficult to really know what are the things I need to include or not. But I'll say, as some of the letter writers are pointing out, we're certainly not getting it 100% right, and are appreciating when people are pointing out where we're making mistakes. But at the same time, I think whatever potential of getting something wrong, if that causes another idea to collide with one, I think it more than pays off.
Yep, totally agree. And I think just one last thought here. Maybe this podcast is pretty far from my wheelhouse, right? But I love it because I'm learning. And you and me both. You can watch me learn, and Nels and I learn in real time, right?
Yeah, that's right.
I think that's fine, and you're going to definitely learn system. The farther you move away from your comfort zone in any area, the better off you're going to be. You're going to learn more. Mm-hmm. And if I had been doing these podcasts from the beginning of my career, boy, I would have published so many more papers. I would have had way more ideas. I don't know that, but I just feel I would have been a better scientist for sure.
Yeah, it's an experiment you can't do, but it's worth thinking about. Yep.
Do you want to finish up these last two here?
Yeah, let's do it. So next letter is from Matt. Matt writes, "Hey, Nels and Vincent, I had a quick comment on episode 23. So that's the same one we were just talking about. The ALUs are going to be all right with the DHX9. In the episode, you mentioned that ADAR enzymes deanimate guanosine to inosine, when in fact, and adenosine deanimases like ADAR deanimate adenosine to inosine, like in the name. The inosine product does look like a guanosine to the translational machinery, though, so you had all the pieces there. The A to G change is then what disrupts the perfect double-strand RNA duplex. Either way, it was a cool story. Thanks again for all of the great podcasts. Best, Matt. P.S. Nels, I won't mention to Brenda that you switched it around." That's great. So there we go. Absolutely. In the heat of the podcasting battle, we
you asked me, Vincent, what does this enzyme do? And I said it's deaminase guanosine, and I got it wrong. And so I fess up to getting it wrong. But of course, it's adenosine. It's in the bloody name of the thing, right? I don't know why I said that. It's probably because I was confusing it, made it look like guanosine or something. But that's great. Thank you for fixing it in a nice way, too.
Yeah.
Right.
Yeah, agree. Thanks, Matt. And also thanks for your discretion with Brenda. So this is Brenda Bass, one of my colleagues here at Utah, who is a pioneer of ADAR biology, some of the RNA biology that we were discussing, actually, which is why that episode ended up being additionally fun for me, is that it's sort of provoked a couple of conversations with Brenda and some ideas that we've been kicking around for a while. And so, this is great. Thank you, man.
All right, our last one is from Steve, who writes, "Hi, Vincent and Nels. I was pleased to hear of Vincent's pick on the way Trump's choices for heads of government agencies are all acting to deregulate the things those agencies were created to control for the sake of people and the planet. As it happens, I've just written a note with an idea about what could be done to prevent this in the future if there was an all-out campaigning effort. There needs to be an expansion of the clear and present danger concept in U.S. rights law. This concept has been used as a way to limit the right of free speech in circumstances when such speech could have disastrous consequences. We need to extend this concept to require speech under the same condition. The Environmental Protection Agency should not be able to prevent its scientists from informing the public about serious consequences of climate change, and the EPA and any other relevant agency should have a duty to inform the public of anything which presents a clear and present danger as determined by reputable scientists. The concept should also be extended to businesses like fossil fuel companies, tobacco companies, chemicals and pesticides companies, and drug companies. With this new constitutional concept in place, situations such as you described and more below would immediately be challenged in court." And he sends a link to a Vanity Fair article, which is basically the same idea as I think it was a Times article that I had originally picked. The Trump White House ramps up its war on science. Days before, 3 scientists were scheduled to discuss the effects of climate change. The EPA abruptly canceled the talks without explanation. That's from this past October, and Steve quotes from that. Nancy Beck has joined the EPA, and she was the person that was being criticized last time in the article that I picked because she's removing some of the regulations, and she was previously at the American Chemistry Council as an executive. So this is, a conflict of interest, and perfluorooctanoic acid is being deregulated when it's really dangerous. So Yep. Thank you, Steve. Steve is in Luton, UK, where it is a beautiful blue and gold autumnal sunset evening on the day the clocks went back to GMT to give us more morning light and less evening.
Sort of a confused emoticon there, or less than happy, bemused maybe emoticon for the time change. Yep.
I think we should stop time changes altogether, but...
Yeah, I have mixed feelings about it too, now that I'm waking up in what feels like the dead of night.
Yeah. All right, that's our letter bag.
Yeah, no, thanks, Steve. Agree that, I would say one thing is that we are putting our democracy under some real selective pressure these days in terms of the kinds of things that we, challenges that we sort of assumed wouldn't happen. And so I'm hoping that some combination of the judicial branch of the government, as well as in the next sets of elections, that we will see what this selective pressure does in terms of a response and things along those lines, because these issues are not going away.
Call your representatives and senators and let them know what you think. It's really important, and it makes a difference. Yeah. Another issue that maybe I'll just lift up for a minute is the new, as the debate, if you call it that, the debate that's happening with the GOP tax cut or tax reform, which appears to involve a pretty serious modification to the way that graduate students are taxed on the tuition waiver, which is calculated on some of the current information and details of the proposals, the House of Representatives proposal in particular, as adding between 100 and 400% to a lot of tax bills of grad students who are obviously not in the greatest financial position as they continue their training beyond their bachelor's degrees. And so that's one, I think, where it makes a lot of sense. I've started to get in contact with my representatives on that one in particular, because I think that's something that could have a massive impact on the way we organize graduate education, not only in science, but
advanced higher education kind of across the board.
There's a very nice Instagram post today from Science March DC, which is a photo of a handwritten calculation by a Columbia grad student, illustrating the devastating impact the bill could have on graduate students. So current tax plan, if you have a stipend of $38,000, you take home $38,000, you pay $4,100 in tax. Under the new bill, you take home the same stipend, but because your tuition is now added to your income, your tax bracket goes from 15% to 25%. Your yearly tax is now nearly $20,000.
It's just crazy.
Pull that out of your stipend. 461%. No, it can't be right. I can't read it. It's too small. 461 increase in taxes. Yeah, it's crazy. I don't understand why you have to do this in order to fund the corporate tax break and the tax break for the very wealthy. I don't get it.
Yeah, no, it seems more like a political strategy, I think, than a fiscal plan that has any kind of logic behind it. And something that's as disruptive of this to graduate education, if you compare that to how little in tax revenue in the whole scheme of things it would save in order to make these decisions, as you're saying, regarding people who probably don't need that kind of money put back into their bank accounts. It's really sort of
indeed
a striking moment in our political life at the right now. And of course, I mean, even if it does go through in some form like this, it would cause universities very quickly to try to adjust their sort of tuition, the way they organize tuition. But even like that kind of a scenario is also a pretty massive disruption that is going to just throw a wrench into sort of how graduate education is organized. And that's not to say that reforms in some ways are worth considering, but I don't know if doing it through this sort of backdoor tax scenario, as opposed to a much more organized and sort of above board conversation, is really healthy for higher education in general, something that's continues to be a massively important pipeline for innovation in the U.S. and everything related to that.
Yeah, it's unfortunate. Well, let's I said this the other day on TWiEVO. Let's become more uplifting with our picks, and then the first pick was a pick about Zika and forgotten babies and mothers. So,
well, I'll see what I can do with my pick of the week here. If you're not nematode like C. elegans, I think you'll be just fine for this pick. If you are one, you might be a little depressed. So I found, came across my Twitter radar, was a video of nematophagous fungi. And so these are fungal strains. Some of them are really interesting just in form and function, that associate with nematodes, including microscopic nematodes, soil nematodes like C. elegans and other worms. And so this video, I did a little digging on it. It's from, I think, the early 90s. It's actually in two parts. It's a bit long, kind of an old school video from some microbiologists at the University of Lund in Sweden, the Department of Microbial Ecology. And they've recorded some really striking light phase video of different fungal strains and pathogens trapping and grabbing worms in different ways, and then building hyphal structures, sort of taking over, infesting the worms, and then completing their replication style. So these fungal strains that hunt. In some cases, about a minute into the first video, you'll see these lasso-like circular structures that constrict and hold the worms in place as the fungus is replicating. It's really a fascinating biology, all kinds of cytoplasmic streaming, and really spicy kind of example of these biological conflicts that are probably unfolding, like in the flower pot right in front of your office plant or something like that.
It's so sad, though. I really feel bad for the worms.
Yeah, they look like they're really kind of vulnerable.
I mean, they're getting these things wrapped around them, and then later on, the stuff attaches to them and sucks out their innards, and then they make spores and they go shooting out of these. Oh my gosh. It's a tough world out there, Nels.
It's wild. That's right. So many ways. I mean, that's right. So many ways to die.
They're just trying to earn a living, these worms, and they can get eaten by fungi. It's a cool video. It really is cool.
Pretty
How
neat.
did you find that? You're just kind of tooling around and ran across it?
Well, no. So I was just, the Twitter feed. I think I have it curated for, folks that are themselves maybe tooling around, or, this sort of network of people just kind of on the hunt for cool biological videos or insights. And so from time to time, maybe even once a day, someone will post something that just kind of catches your attention. And this is the most recent of them. So it's really fun to see.
Cool. How about you? There's a guy over at your place, Ed Grow, who does a lot of cool retweeting on Twitter.
Yeah, I agree.
Follow him, yeah.
Yeah, I do. He's a postdoc in Brad Cairns' lab here. Yep. Okay. Also a good, faithful listener of TWiEVO, one of our regulars. Good guy. How about you, Vincent? What's your pick of the week?
So I have an article in eLife, which is an open access journal, so you can all go and read it. It's called "How Should Novelty Be Valued in Science?" And the idea here is that scientists are under increasing pressure to do novel research. So the author Barak Cohen of Washington University School of Medicine explores whether there are risks to overemphasizing novelty when deciding what's good science. And the article reviews the philosophy of science, to understand how an emphasis on novelty might be important. He reviews studies from sociology of science, and the conclusion is that placing too much value on novelty could have counterproductive effects on both the rate of progress in science and the organization of scientific community. And he has a suggestion that our current emphasis on novelty should be replaced by a renewed emphasis on predictive power as a characteristic of good science. I'm not sure I agree with that. Mm-hmm. I think it's very hard to predict what's happening or what's going to result from any aspect of science, right? You say, here's CRISPR, I predict this is going to allow genome editing. You had no idea.
Yeah, right. Yeah, true. But I do think maybe some balance in it, even if it's not the same person or the same research program, if one group is pushing into, crazy genetic structures that resemble CRISPR or was CRISPR 15 years ago, and other folks are doing modeling or, work to sort of bring some predictive insights into sort of how a biological process or any scientific sort of process is organized, that there's value in both.
Yeah.
I did see, I have seen studies where just people, will sort of track, and you can do this on Google as well, where you just track the prevalence of a word in, in this case, in scientific articles. Mm-hmm. And the word novelty, I think, has had an especially drastic rise in the last 30 or 40 years, where people would almost never use the word novel in a manuscript, whereas now it's all over the place. And, even in titles, the word novel is almost like has become almost a generic throwaway word because it's used so commonly. So there's been a sort of inflation on the word novel, and I think that probably echoes in some of those ways as well, as people think about what they're looking at.
I don't know. I think you should just identify good scientists with good ideas and let them do what they want. That's the way I think it should go. You fund them. If they don't come up with anything, if they don't publish anything, all right, that's the end. But you just don't know what's going to be novel or what's going to be important and what's going to happen. And just let smart people do their science. And that's the way it was. And I think that is the best way to get your money's worth.
Mm-hmm. Agree. Yep.
We have a couple of listener picks.
Great. So I'll do the first one here. This is John. He writes, "Hi, Vincent and Nels. I know that someone on the TWiCS teams picked a previous a cappella science song about CRISPR-Cas9 as a pick of the week. This one is new, and I figured it might fit on TWiEVO." This is a great one. This is the Evo Devo to the song Despacito, the very ubiquitous song that had a tremendous rise. And yeah, this is, John, this is awesome. I saw this a while ago. Again, I think it came across my Twitter radar. and a really spectacular, inspiring, lively video to sort of lay a foundation or an introduction to the whole field of Evo-Devo, evolutionary developmental biology. I would definitely recommend this to all our listeners, if they haven't seen it already, to take a look. I think it's had a pretty good run already. It's gotten some really nice attention.
Yeah, it's cool. I love it.
Yep.
Just very
Enjoy.
very
Thanks
clever.
in regards, John in Limerick. Yeah, keep an eye on this acapella science. I think it's really developing in cool ways.
All right, our other pick is from Melanie, who writes, Dear Doctors Elde and Racaniello, I want to thank you for your dedication to this podcast. As a scientist surrounded by non-scientists, it is often the only thing that makes me feel not so lonely. I'm a biologist working for the federal government, and sometimes I feel like an iceberg in the Caribbean, out of place and slowly breaking up. The TWiEVO podcasts really help keep it up. I'd like to suggest a pick of the week. Most of what you guys talk about is on the gene or molecular level. How about looking at evolution through comparative anatomy? What's in John's Freezer is an informative and fun site for anyone interested in studying evolution on the macro level. Keeping this short so you can get back to podcasting. So this is a website called What's in John's Freezer, and this is a fellow in I guess he's in the UK somewhere, who has a collected
in Ireland. Yep.
Ireland.
Mm-hmm.
He's collected. Yeah, I can't say the UK, but the Irish get mad when I do that.
Yeah.
He's collected things in his freezer, and he has a consensus here. So far, he has 475,325 things in his freezer. And so he periodically puts up pictures, and, you can see what's in there. And I was thinking today, as we were doing today's, maybe you could pull something out and sequence its genome, right?
Yeah, that's right. Then we're right back, though, to the genetic and molecular level.
Yeah.
No, it is true. I mean, there's so much great evolution and ecology and evolution as well that, now that we can dedicate ourselves, now that we're through our first quarter century of podcasts, TWiEVO 25. I can guarantee that we'll continue with a lot of stuff happening at the molecular and microbiological levels, but certainly including some more kind of ecology and some macro level stuff is a really great idea. So in John's freezer, what's in John's freezer? It's a week or two ago, but I was kind of tooling around there. A really cool, great energy and interesting blog.
It's really nice
to look at.
It's really
yep.
Nice.
Yeah. So thanks, Melanie. Glad we're able to keep you some company there, and keep listening.
All right. Well, thanks for those picks. Send them in, and you can find TWiEVO at Apple Podcasts, microbe.tv/TWiEVO. And if you like what we do, consider supporting us financially. If you gave $1 a month, that would be cool. Help pay the bills. Microbe.tv/contribute. And send your questions, comments, picks to TWiEVO at microbe.tv. You can find Nels Elde at cellvolution.org. You can also find him on Twitter. He's L Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Good to dive into the mailbag a little bit here, and good to kick around a really interesting and provocative paper.
Always. I'm Vincent Racaniello. You can find me at virology.ws. Music you hear in TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. Been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, be curious.